Hi everyone, I am Saniya Khullar. I am the TA for the bioinformatics class, and today I want to go over an example on Gibbs sampling. For those of you who are wanting to go over the concept and have some questions about it for the homework as well, I think that this would be very helpful to go over how do we use Gibbs sampling. So I'm going to go through a simplified example of Gibbs sampling right now. So in this sample problem, please suppose that you're running the Gibbs sampling algorithm for motif finding on a set of 4 input sequences with a motif width of 4 nucleotides. So this has to do with like transcription factors, recognizing binding sites along the DNA sequences, which is super important, especially because like whenever we have transcription factors, they usually try to bind to sequence-specific DNA sequences. So if you see it here, like in this example, for instance, if you have a transcription factor, these are typically proteins, and they help regulate the expression of genes, the activation or repression by binding to the sequence-specific regions. So these are regulatory elements like promoters and enhancers, and they're found in the non-coding regions of the genome. And so transcription factors bind to these sites, and they help recruit RNA polymerase II, which helps transcribe the gene. So if you think of these transcription factors here as being over here, for instance, this is a transcription factor, and this is the DNA sequence specific binding site here that this transcription factor will bind to, much like puzzle pieces. So you can imagine this transcription factor has TAC, GTAC here, and it's going to bind to the sequence specific motif on this transcription factor binding site, A, T with A with T, C with G, G with C again, and they all just bind here. And when it binds here, it's then able to help regulate the expression of this gene. So the understanding of these motifs that they bind to, if you look along a sequence of the DNA, it's really important. And there are many different techniques that can help us figure these out. And one of them is Gibbs sampling. So please suppose that you're running the Gibbs sampling algorithm for motif finding on a set of 4. So this is a toy example of 4 input sequences with a motif width of 4 nucleotides. And please suppose that the current state of the Gibbs sampling is as shown below, with the motif occurrences indicated by the underlying nucleotides. So basically, wherever we see these underlines here, this means that this is the motif. So here, so we have these 4 sequences here, and we think the motif is here, like GTGA in sequence 1, AGCA in sequence 2, CTCT in sequence 3, and AGAC in sequence 4. So please suppose that in the next iteration of the sampler, we pick out sequence 3, and the motif position in that sequence is to be resampled. So we're gonna resample and figure out the best motif. So we start out with believing that these are the motifs. And like, so we start out believing these are the motifs, and we're just going to resample the 3rd sequence. So please compute the parameter estimates P. So this is like the probability weight matrix that are calculated in the predictive update step, given that sequence 3 has been selected. Please use a pseudo count of 1 for your estimates, and assume that a stranded probability weighted matrix model that does not incorporate any prior knowledge. Please be sure to give the estimates for both the PWN and background parameters. So when we have a pseudo count of 1 basically means that we want to believe that each base has some probability of being there, even if we're not so sure about what the exact probability is. So why is this important, for instance? Well, it is important because, for instance, a lot of these, if we look here, for instance, if you look along the genome, we can see that this is TGCA. We can see that like this is like a motif, for instance, that a transcription factor like REST binds to along the regulatory region here on this chromosome. And we want to figure out what is the motif look like what's the probability of having, so these are related in some way to the probability of a base being in that certain position. So we see here that this is really strongly, almost pretty much 100% likely to be an A. Here it looks like more equal. So the more equal it is, generally the less information content you're going to have in each of these locations here. So if it's more probable that it's A, like that's the real base, so you see it's like super high because it's super likely that it's just one base. And same here for G, it's like really high. But here, position 3, it's like a C, and then you can see it can be a T as well to some extent. So it's sort of like sharing some of the thunder with T. And that's why it's less in height. So the more like certain we are that it is that base in that location, the higher the sequence will be in the sequence logo, that base will be in the sequence logo. So we start out with, and we're focused on all of these, we're focused on doing this predictive update step for sequence 3. So we're going to just take this sequence 3 out, and we're just going to focus on sequences 1, 2, and 4. And again, these are the current motifs that we have that we're going to use to construct our probability weighted matrix. So please note that we fix these other sequences 1, 2, and 4, and then compute the P matrix only using these 3 sequences. So since sequence 3 can change, we will not use it to compute the values for the P matrix. So in this Gibbs sampling method right now, we are going to be using sequence 3 and doing updates on that. So again, we're focused on these. So the first thing that you do when you have this set of sequences, like here with these current motifs. So again, we're focused on updating sequence 3. So we do not consider it, and we look at sequences 1, 2, and 4. So the first thing that we're going to do is for these 3 sequences, we're going to find the counts of bases in different positions across the motifs in these, these should be these 3 different sequences. So we have 1, 2, 3, and 4, because each of these is a motif here, and it has like 4 positions because the width is 4. So we're going to look at each of these, and we're going to try to find for A, C, G, and T, how many out of these in these 3 motifs, how many of the first positions are A's? That's what this is going to tell us. How many of the first positions are C's? That's what this will tell us. How many are G's and how many are T's? So right off the bat, you're going to see that they can be either G's or A's here. So we should have nothing for this cell here and nothing for this. Similarly, what this K equals to, this is the position in the motif. It's going to tell us that how many A's are we going to have in the second spot in the motif. So we can see here we have none. So it should be 0 for A, no A's and no C's actually. So we're going to look at this and we're going to fill out. So similarly, this tells us the number of C bases in the second position, number of G bases, number of T bases. So we're going to scan through all these 3 sequences and add those up. And that's what we want here. We want to get the number of each of these bases in the different positions along the motif. So right off the bat, if we want to look at the number of A bases in the 4th position of the motifs, we look here and we look in this 4th position here. And we're going to say, hey, this is an A and this is an A. So this number should be a 2 over here. So it should be, this should be a 2. That's what it will be. Similarly, for the C, we're going to say, oh, we actually have 1 C over here. So this would actually be 1. So this would be 1. And we have no G's in this 4th position here. And we have no T. So those should be 0. And this should also be 0. So I'm going to break it apart again. So again, what this corresponds to is we see here, we take these sequences and we look at them across sequence 1, 2, and 4. So G, T, G, A. And this is again how we can break them apart, A, G, C, A, and A, G, A, C. So what we're going to do now is we're going to then take the number of A bases in the first position of the motif. It's going to be 2 because we have 2 of these A's here. Number of C's is going to be 0. Number of G's is going to be 1. Number of T's will actually be 0 in this first position. And similarly, we're going to look here and we're going to see a number of A's as 0. So we're just going to count for each of these bases, we're just going to sum up the number of times that they appear in this respective column for K. Again, this is sort of how you can juxtapose this for this Gibbs sampling problem. Sorry, they should be 3. There's really 3 here. So again, what you'll see here is that I've just had this become 2 for the A here. So the 2 times that it appears at these positions along the motif, this is a 2. For A, 0 for C, 1 for G, and 0 for the T. So similarly, we find that there's 0 A's in this second position. There's 0 C's in the second position, but there are 2 G's in this position. We find that there's 1 T in this second position. We also find that we have 1 A in this third position, 1 C in this third position. We also have 1 G in this third position, but we do not have any T, so this is 0. And we have like 2 of these A's here, so this is 2. We have 1 C, and we do not have any G's or T's here. And when we add these up, these are 3 as they should be because we have 3 sequences. So of course, we will, the number of first positions would be 3, of second positions is 3 and 3 and 3. So this should add up to 3. And then basically, this is the notation that we learned as well in class. So this is telling us the number of A bases that is in the first position. That's essentially what this is, a number of A bases in the first position of the motif. And that's what K is measuring, the position that we are in the motif. So whenever K is 0, that's the background. We were going to get to that next. But this is trying to calculate like how many A's are in this first position, how many C's are in the first position, how many G's are in the first position, and how many T's. And for whatever reason, in this whole sequence, we were given that these are the motifs, these underlying parts. So these underlying parts are what we believe are currently the motifs. And that's sort of like what we're working with right now, but we're focused on updating this layered sequence, but we are given to work with these motifs for now. And again, so what these values here are, these correspond to what these actually stand for, and they add to 3. I just put the notation in here. So in Gibbs sampling, like, you know, we're going to focus on, these are the key variables that we focus on. That PCK, it's a probability of character C in position K, where K is the background non-motif, and K is greater than 1 represents positions in the motif. So pretty much what we want in a probability weighted matrix is we want to find the probability of each of these positions taking place. So for instance, what you're going to see is like in the homework assignment results, what you find is that we are often given a probability weighted matrix where this is our background. So this is our background. This first column is our background. So this is pretty much our K equals 0. So I'll just write it on top, K equals 0. And this is like position 1. Oh, sorry, this is K equals 0. Position 1, position 2, like if we had a motif, and this is position 3, position 4, position 5, and then this should be position 6. So these over here are all like this part here. This is all like motif, the motif. And what this is this is a probability weighted matrix where you see that all of them sum up to 1. And this is telling us, so this is one of the example ones from the homework as well, where we're finding out using the EM algorithm, we're trying to figure out like, what is this, using mean. We're trying to figure out like what is the corresponding best probability weighted matrix. So similarly, we look here and we find that the highest background is for base C, then like for the background, but it's like pretty much equal. It should be around 25%. But you know, it may vary, you know, like the non-motif bases. But then we look inside the motif and we see in position 1 in this example, we are really likely to have base C here. And then next is T. So like very like C and then T's, we would expect a lot more. So this is like a 43.1% probability that the first position in our motif is a C. and a 38.1% probability that the like the position 1 in the motif is a T. And similarly here, for instance, it says that a 53.2% chance that we have an A in the second position in our motif, and then next it's met with a T, which is 42.4%. So we can see here that these all add up to 1. This is an overwhelming amount of T's in the third position of the motif with 87.8% probability. And here it's almost pretty much certain, like for these fourth and fifth positions of the motif with a 92.2% probability and 93.5% probability of having a C in positions 4 and 5. And here it's like roughly like, you know, majority is G, but then it also has some T probability. So this is what a probability weighted matrix is. And we want to be able to figure it out while holding out sequence 3. We want to work with sequences 1, 2, and 4 to find what this corresponding probability weighted matrix is. Because as you see, these are all like probabilities essentially. So whenever these values are greater than 1, greater than or equal to 1, we are looking at positions in the motif. That's what we want to go from these n values all the way to our probability values. And we are going to do that with this formula, that we're going to find the probability of having a given character in position K of our motif, like in positions 1, 2, 3, or 4. We're going to figure this out by using this motif probability, which is n c comma k plus dc divided by n minus 1 plus db. So you can think of this n minus 1 is just, you know, it should be in like, you know, like you just have to subtract n minus 1 and then add db to it. I should have made that clearer. So this db is a pseudo count because we want to make sure, and this also is a pseudo count for the numerator, because we don't want any zero probabilities for our PWM. We want it to pretty much be, in this case, we want it to be smooth. It's like something that's known as Laplace smoothing. We want to make sure that we have at least some value, even if it's like really tiny. We just don't want a zero probability for a base, because who knows, there can always be some probability that the base is something. So we just add 1 to it. And since we're adding 1 for each base, we also add 4, like so 1 plus 1 plus 1 plus 1 to the denominator. And this is something you'll see in like natural language processing, for instance, when you're trying to figure out, or using naive Bayes to figure out like do classification tasks. So what you see here is we have n, total number of sequences. So to begin with, we had 4 sequences in this data set. So it's a toy example. So n equals 4. But we're using 3 of them because we're holding out sequence 3, but in reality, we just have like 4 total sequences. The sequence length is the 8, L equals 8. And why is it 8? It's because in this original sequence, we have 1, 2, 3, 4, 5, 6, 7, 8. 8 is the length. That's how we know that it's 8. The sequence length is 8. Then we have the pseudo count for a character, and a single base is 1, as I mentioned. So this is to fluff it up and make sure we don't have any zero probabilities. And this pseudo count for all 4 bases will be 4. Our motif length is 4 as well, because again, for each of these motifs, we just have these 4 bases. So that's what we're looking at. So for the motif probability, what we do is we have this NCK, and then we add 1 because this is what DC is. And then we have 4 minus 1. Again, the way you can think of it is 4 minus 1, which is 3, plus 4, which is 7. So throughout our denominator is going to be 7. And so I simplified this motif probability down to what we're working with when we're trying to find the probability of a base in a motif. So it's like us trying to calculate essentially, what is this? When we want to find out, like we're focused on each of these. This is essentially what this is saying, is that probability of C being in position 2 of the motif is equal to 0.431. So what that would be is this would correspond to essentially saying that P, you know, like if this is a subsign C comma 2 is equal to 0.431. Then similarly, like if I were to look here, this would be T in position 3 of the motif. So the way I would interpret it is T being in position 3 of the motif is going to be 0.878. And what that is this is t comma 3 equals 0.878. And that's how we interpret it. So it's p. Actually, I can also make these like really small because that's how it looks here. That's how it looks. T comma 3 0.878. So the probability in this case that the first motif position base is an A, I'm going to go back and I'm going to use this table here to figure out what is that exactly. So as you see here, that I'm trying to use this formula now to find for each of these positions. So I'm going to go through and I'm going to try to convert all of these to probabilities. Essentially, what this involves is just me adding 1 to these numerator values and then just dividing by 7. That's essentially what it does. So if we had this in Excel, this is the same thing as me adding 1 to all these values and then just dividing it by 7. So similarly, what this is it's just me taking an A comma 1. So the number of times I found an A in position 1 of the motif, I add 1 to it and then I divide it by 7. So I get 2, which is what this is, 2 plus 1 over 7. So that's 3 over 7, which is about like 42.86%. Similarly, what I do is, let me just bring this guy down here a little bit so we can see that this is the same. I do the same thing for C comma 1, and that's how I get my 14.23% here. You can see here that it's there's no counts of C, so it's 0. 0 plus 1 divided by 7, 14.23 here. For G, there's 1, so I get 1 plus 1. So this is me adding the pseudo count again, and this is 2 over 7. And again, I'm using this formula of NCK plus 1 divided by 7, and I got it from here using these, that's nice. So what we also see here for T, there's nothing, but you can see this is a reason why we have the 1 over 7, because we don't want it to be a 0 probability. Even if it's small, we still want to collect that. So this is 14.293. So we add this 1 as well. So just to give it some chance as well, because we want to also account for the fact that we only have like such few sequences, right? Maybe there could be a T. We just don't want to say that there's no chance whatsoever. So we do the same thing again next for the second position. So now we want to see what is the chance that the second position of a motif is an A. So we're going to go back to this table and we're going to say, okay, well, we see here that A's and C's are 0. So we would expect the same thing where this is a 0. And again, it's 14.293. C is also a 0. For G, what we see here, again, bringing it back here, is that we have about 2 bases here that are G and one that's like the pseudo count. So it's 3 over 7. And again, we got these values because what we did is we looked here for these and we saw that how many times do we find a G in the position 2 of the motif? We found 2 of them. That's how we'd gotten that count of 2. And we found 1 T, but we did not find any A's or C's. That's why these were 0. And these were 0 here. And that's why we couldn't find those counts there. So, and again, like the multiple ways of doing this, I just think this is a nice standard framework to use. So again, we also look in the third position as well. We find these counts, oh gosh. So we look here and we find these counts. In the third count is a T, for instance, and we find that there's none here. But for the G, the third base is a G, what we do is we find that there are 2 out of the 7. So we take 1 plus 1 because it's one G in the third position. And then we just add 1 to it, and then we get 2 out of 7 here for G being in the third position. And then in the fourth position, again, we see that we have 2 A's for NA comma 4. So we put 2 plus 1 and we get 3 over 7. And similarly, we get like, we can find the C's is 1 plus 1 over 7, so 2 over 7. And this is sort of showing again the effects of the smoothing with the pseudo counts of 1 that we get like this 15.38. Actually, I think that should be, oh, sorry, this is my bad. There should be a 14 point. 0.285. Sorry, my bad. 14.285. Let me just move him here. It should be the same here, roughly. So and then for the background probabilities, what we do is we use this formula. So again, now what we're trying to do is we're trying to figure out what are these probabilities for the background. Usually our assumptions are that it's like 0.25, but what we want to do is we want to figure out like what is the chance that like it's like an A, G, C, T. Is it like really different from 0.25? Like what could it be? Maybe there's just a ton in this area of C's, or who knows, right? But what we use is the same idea here that I mentioned, the same formula from above that we had used is what we use here. Like this formula here, pseudo counts and W. I just brought it down for our reference. So what we see here for the background is we want to find if it's not a motif, what are the probabilities of, what do the bases look like? So this is the formula that you actually use for the background non-motif probability. And we will use NCO is the probability of having a given base in position zero of the motif. That's what this probability is. And this N is the number of times we found that base in position zero. That means in a non-motif position. So we're trying to find the probability of us observing a particular base in a non-motif position. Like, so if we're not looking at motifs, like usually what bases do we expect? So what this is telling me, for instance, is that I would expect like 27.5% of the time that in non-motif positions, I would find like a base C. 22% of the time I would find a G in this background position, like non-motif. And then 25.2% time, it'll be a T and an A as well. So that's what it tells me. So that's, these numbers are the counts. And then this DC is the pseudo count that I'm adding. This is N-1. This is the length again, which is 8. And this is the width, which is 4. So this is the probability. So I'm plugging it in so I can just calculate these values and I don't have to worry about them again as much. So what we find that this is the background probability again. So what this PC comma 0 essentially is saying is it's what this essentially is again, is the probability of observing a base C in position. So actually you can call it in the background position. So you can think of the like non-motif as like being a background. It's just like something that's happening behind the scenes in a way. So that's what this is. So non-motif, like if in the background, if we didn't have a motif, what do things look like? So this is like ATGT, this is CAGG, and then this is CAGA. These were the motifs we identified. So before, again, these were what we had identified as our motifs. And now everything else is the background. Like, so everything else is kind of happening behind the scenes. It's not the motif, right, for these 3 sequences. So whatever remains will be the background. So these are the background. So the CATG, this is the part that we ignored. This is the background for sequence 4. This is the background. And again, we only look at these sequences 1, 2, and 4 because we are holding out sequence 3. This is the C here. And then this is the GAA here. So we are holding out everything else. These red are the backgrounds because these are not motifs. These are the motifs as the CAGA. You know, we had the motifs as, you know, CAGG, and what's left is this part is non-motif and this part is non-motif. So it's a background. This is also background, and this is background, and this is background as well. So what happens is that essentially we just take away the motifs and we look at the remaining bases. So we just focus on what's highlighted here. And please only look at the background bases for each of the 3 sequences. So this is, and again, there can be more. Just for this example, there's 8, so it's like 4 and 4. But in a way, this is like CGAA. I just smushed together everything that is a background. This is CAGG in the second one, and then this is CATG. So now I'm going to do somewhat really similar to what we did before, like for the motif positions when we had like all of these and we were trying to figure out again, like we were looking at, I think here, yeah, we were looking at like G, T, G, A, like I was trying to find the counts in each position. This is pretty much the same idea, where what we're trying to do, guys, is that we're just trying to figure this out, where for the background, it looks like this, where I have the CG. So these again, these are the backgrounds. After I subtract the motif parts, I see this is CGAA, CAGG, CATG. This would be 2, 1, 1, and then 0, because there's no base 0 in the sequence. There's no base T in the first sequence. Here, that's 1, 1, 2, and then 0 in the second. And then we see here that this is 1, 1, 1, 1. It's got 1 of everything. And then just to make sure, so we can add these across as well, we would expect to see 4 here, because in this, we have 4 total that are A's. So we expect this to be 4. And then this would just be 3 again. You know, we find that there are 3 C's, 1, 2, and 3. Actually, it starts out with C. So it's pretty much, I would expect that in the background, this would almost be like, you know, very high. I don't know if this is a motif, this would be 100% for position 1 in this case, but you know, it's not a motif, it's a background. So NC 0 is saying that we found 3. So again, don't get confused. So in the background, you do not worry about these positions at all. All that's saying is once you're in the background, you are in the background. Everything is basically a position 0. It's just, we're figuring out if you're not in a motif, if you're just all these red bases. So we really worry about positions for the motif because the motifs are really specific. If you're not in a motif, like these red bases here or these highlighted yellows, we're just trying to figure out like what's the probability you're going to be a certain base. So the position doesn't matter. So for here, all we're focused on is we're just focused on counting the number of times we see a given base in a background. The position does not matter. So here we see 1 T here, so that will be 1. We see 4 G's here, so this should be 4. We see 3 C's, so this should be 3. So again, now we just use these same counts and we use this formula here of this probability that I derived here. And we're going to find what this probability is. So the probability that the background base is an A will be equal to, we take this and we add 1, we divide by 16 here, and we get 4 plus 1 over 16, 5 over 16, which is 31.25%. And similarly for PC of 0, we'll get 4 over 16. You know, we take 3 plus 1. We get 4 over 16, which is 25%. So 31.25% of the time, we're going to find that, you know, it's going to be an A here. And it's not like you could look at this, but again, you also have to make sure you add 1 to the numerator and, you know, you add the pseudo counts to the numerator and the denominator. So it's like the probability that you observe the bases in these sequences, but you've also added some smoothing. You've added some value to the numerator and to the denominator too. You've done some adjusting. So that's what it is. So, and pretty much what does this mean exactly? So this is just saying that, what does this formula mean? So all that's saying is that, and it makes sense because all we're saying is we're counting the number of times we observe a given base over the total number of bases that there would be. So that's what, so if you think of what a probability conceptually means here, this is what a probability would mean to us. Like if I asked you, what is the probability of us observing an A in the backgrounds here? Like, you know, oh gosh, an A in the backgrounds. What you would say is that, oh, okay, so I see that we have actually 1, 2, 3, 4. So I would say, okay, we find like that we have like 4 A's in background. So like in non-motif regions, it doesn't matter again, those positions in a non-motif. It only matters for the motif. So we have 4 A's here. And I think that there's 4 bases plus 4 plus 4. So that's like 4 times 3, right? So 4 times 3. So that would be 12. So I would say that the probability of observing an A in non-motif would be 4 divided by 12, which is about, you know, 1/3, which is about 33.33%. That's the probability I would expect, right, for an A. But what this smoothing also does is it's trying to also like balance things out, because some things might have 0 counts. And again, this is what smoothing does. So what this formula is, all it's saying is that we have 3 sequences. So that's what the first part is saying. We have like 3 sequences. So 4 minus 1, which is 3. And what this is saying is you find the length of these sequences, which should all be the same length, and you subtract what the length of the motif is, and that's just giving you the length of the number of bases that are in non-motifs. So this is just 4, so 4 in motifs in each sequence, 4 non-motifs in this example. So that's what it is. It's just like you take this and you divide it by 12. And that's how we'd gotten that number above here. I'd gotten this 12 that way. Like this NC0, like n this 12, this is pretty much what we expect. And again, this NC0 is the number of bases in background. It's a number. That's what this NC0 is. It's just a number of C bases. C bases. And what C is basically, in this case, in this alphabet of the DNA, C is the way that you can think of it as C is inside this list of A's, G's, T's, and C's. That's what it is. And we're looking for each of them, we can find those. So this part here, like this part NC0, this part here in red, and this part is what a typical probability is, what you would expect. And what this 1 and this 4 are is these are pseudo counts. This is like your smoothing again. So again, so don't be worried. This is what you would naturally think of as probability, but we're just adding a smoothing to it. And that's how we get like this 5/16. We've just taken 4 A's out of 12 bases plus the smoothing, which is 4/16. And then this is like 25% of A. And you can see here that this smoothing kind of levels things out, because we would expect this to have been 33%, but with smoothing, it's kind of pulled down a little bit to 31.25%. Meanwhile, other ones that are lower would get pulled up a bit more. Like you can see like this T probably would get pulled up a little bit because we would find this 1/12, which would be 1/12 is approximately like, I think 8.33%, and this 1/12 background bases, but it gets pulled up because of this to 1/8. It gets pumped up by 1, and it's 12.5%. So it got bumped up from 8.3% to 12.5% with the smoothing. So this is again how you find it. You plug in this formula, you take these counts, you add 1 to it, and you have 4/16. This should be 16 as well. This should be 5/16, my bad. This should be 5/16. And then this is like out of 16, this is 1/8. So in the end, we have this background base in the DNA sequence, and these are the probabilities that the base could be one of the respective DNA bases. So if we're looking at a background base that's non-motif, background, I can have non-motif. It's not a motif. So we either have motifs or we have background bases. So 31.25% of the time, it's going to be an A. 31.25% of the time, it's also going to be a G. So they were tied there. And T's and is going to be 12.5%, and C's are going to be 25%. So these would all add up to 100%. So what you're going to observe, hopefully, is that and that makes sense, right? Because this is 1, it's the lowest, and these are 4, so we have the same results for these, and then this is slightly lower because it's a 3. So what we do next is, I just had to make this small change here and here. So I think, sorry about that. So what we do then is we just go back and we put in these probabilities here. So again, what this is like PA of 0. This is PC of 0. Oh gosh, actually this is better. Maybe I should do it this way. And then I'll just fill it in for you guys. This is PG. This is a probability weighted matrix. And in the end, this is what these values are that we've calculated above. We just plug them in from above and we get our results. So this is T0, this is T1, this is T2, T3, T4. So this is sort of like how we go about it. And we just plug in these values, and we have computed all these values from above. So I just plug them in and we're ready to go then. Oh gosh, and this is it. So that's how we have all of these values in the probability weighted matrix. So probability weighted matrix if we for the Gibbs sampling. This is for Gibbs sampling, where we are pulling out sequence 3. And this is what we get for the probability weighted matrix. And again, these are all these values that we've calculated throughout. And this is like all the whole step, we've been going throughout calculating all these probabilities here throughout. I've walked through how we calculate all of these, going in reverse as well, starting out again, like in a motif, the positions matter. And we want to figure out, because we want the motif, but in the background also, it's not a motif. So it can be as arbitrary as possible. The position doesn't matter because, I could have my motif at the beginning or end. It doesn't matter. Like all this comes a bit afterwards or before or in between. Like it doesn't matter. So the background is the background. We just want like the probabilities that we observe certain bases in the background. It's the motifs that we really care about, what those positions look like. So I hope that this makes more sense about how you go about like the Gibbs sampler. So please let me know if you have any questions on this. Thank you guys.